Mechanism of hearing. Ear is divided into three parts: external ear, middle ear, and inner ear. External ear has got two parts: pinna and external auditory canal. Here we can see this is pinna, this is external auditory meatus, this is external auditory canal. This forms the external ear. The pinna. The pinna is a cartilaginous expanded portion covered with skin projecting from the side of the head. The functions of pinna is to collect sound waves and localize the source of sound from where it is coming. Secondly, it funnels the sound waves into the auditory canal so that the sound wave can reach to the tympanic membrane. Now, role of external ear in hearing. The external auditory canal is 2.5 centimeter long, runs forward and inwards, that is in medially. It extends from pinna to the tympanic membrane. It is lined with the skin and that contains glands like ceruminous glands and sebaceous glands. Ceruminous glands secrete wax and sebaceous glands secretes oil. The functions of auditory canal is to help in transporting the sound waves to the middle ear, and the earwax and hair prevents the entry of dust and foreign bodies into the ear. Now, role of a middle ear. Middle ear consists of tympanic membrane, ear or auditory ossicles, pharyngotympanic or eustachian tube, and there are two skeletal muscles present called as tensor tympani and stapedius muscles. Now, tympanic membrane. Tympanic membrane, it consists of connective tissue covered with the skin on the outer portion, and inner portion is covered by the mucous membrane. It is a funnel shape with center umbo pointing inwards and is kept in position by the handle of malleus. The functions of tympanic membrane is it acts as a pressure receiver, resonator, and dampens the sound waves. The pressure receiver is that tympanic membrane is very sensitive to pressure changes produced by the sound wave on its external surface. Resonator means that tympanic membrane starts vibrating freely when the sound waves strike. Damping can be made to understand is that it stops the vibration of the sound waves. This means that the stretching of the sound waves, the tympanic membrane stops immediately. That means it dampens the sound vibrations. Ear ossicles. Here, ear ossicles or auditory ossicles. The function is act as a lever system that converts the resonant vibrations of the tympanic membrane into the movements of stapes against the parallel scala vestibuli of the cochlea. Here we can see that this is malleus, incus, and stapes. These are three auditory ossicles. Middle ear contains air and the inner ear contains fluid. So therefore, the sound has to be transmitted from the ear to the fluid. And fluid has got inertia. Therefore, for the transmission of the sound into the inner ear, the pressure is to be increased in the middle ear so that the wave can be transmitted from the middle ear to the inner ear or from air to fluid medium. Factors responsible to increase the magnitude or pressure in the middle ear. These are the factors. Ear ossicle act as a single unit. Now we can see that the size of the handle of malleus is less than the size of incus. This is handle of malleus. The size of the handle of malleus is less than the size of the incus, and the size of the stapes is less than the size of the incus. and the long handle of malleus is longer than the long process of the incus. Handle of malleus is longer than the long process of malleus. So these are the factors that leads to the increase in the magnification of the sound intensity by 1.2 to 1.3 times. Now, second factor is the surface area of the tympanic membrane is 50 millimeters square, and that of the oval window is 3 millimeters square. Thus, the reduction of the size is about by 17 times at the level of the oval window. This results in corresponding increase in the pressure at the oval window. So, these both factors increases the pressure within the middle ear by 22 times, and this is referred as impedance matching. Thus, when the steppiece is pressed into the oval window, this pressure is transferred to the perilymph in the scalar vestibuli of the internal ear. The role of two skeletal muscles is to provide a protective reflex called as tympanic reflex or acoustic reflex. The last sound initiates a reflex contraction of the middle ear muscles. Whenever it's a protective reflex against the loud sound, and when the loud sounds are produced, or it will initiate the reflex contraction of the muscles. The muscles that are contraction of tensor tympani muscle causes tensioning of the tympanic membrane, leading to the decrease in its vibration, and contraction of the stapedius muscle leads to the pulling of the foot plate of the stapes out of the oval window. Thus, the contraction of these two muscles prevents excessive sound going to the oval window. Loud sound can lead to the excessive displacement of hair cells from the tectorial membrane, which results in deafness. Now, the role of inner ear. The inner or internal ear consists of two parts: bony labyrinth, and membranous labyrinth. The bony labyrinth is a series of channels in the temporal bone which encloses the membranous labyrinth. Membranous labyrinth comprises of one vestibule, one cochlea, and three semicircular canal. See, here we can see three semicircular canal, cochlea, and vestibule. Vestibule contains utricle and saccule. Now the cochlea is an organ of hearing, and it is responsible for hearing. It is a snail-shaped coiled structure, coiled tube, 35 mm long, and which makes about 2.5 to 3 turns. It has an outer bony spiral canal that houses an inner membranous cochlear duct. The membranous cochlea or cochlear duct is filled with endolymph. See, here we can see this is external ear consisting of external auditory canal. Here is a pinna. Then this is beginning of the middle ear. This is tympanic membrane. Auditory ossicles, malleus, incus, stapes, and this is oval window. Foot plate of the stapes lies over the oval window, and this is round window covered with secondary tympanic membrane. The thing is, the cochlea is divided into compartments by the Richner's membrane and basilar membrane. So these are the three compartments: scala vestibuli, scala media, and scala tympani. Right. The scala vestibuli is a space above the Richner membrane, and it is filled with perilymph. Here we can see this is the Richner membrane. Richner's membrane that divides scala vestibuli from scala media. Scala vestibuli contains perilymph. This is filled with perilymph. Scala media contains endolymph. And thirdly, basilar membrane. This is basilar membrane that divides scala media from scala tympani. Scala media, as we know, that contains endolymph, and scala tympani contains perilymph. So here we can see the space is divided by Richner's membrane. This is diagrammatic sketch. Uh, this is divided. This is Richner membrane, and this is basilar membrane, and divides into three compartments. Scala vestibuli filled with perilymph. Scala media is filled with endolymph. Scala tympani is filled with perilymph. So scala vestibuli is the space above the Richner membrane filled with perilymph. Scala media is a space between the Richner membrane and basilar membrane and is filled with endolymph. Scala tympani is a space below the basilar membrane filled with perilymph. At the apex of the cochlea, the scala tympani and scala vestibuli communicate by a small opening called helicotrema. See, at the apex, the scala vestibuli and scala tympani is connected to each other by a small opening that is called as helicotrema. At the base of cochlea, the vestibuli, scala vestibuli ends at the oval window. The scala tympani ends at the round window. See, we can see the example here. The scala vestibuli, this is apex of the cochlea. This is apex of the cochlea. This is base of the cochlea. Apex is connects scala vestibuli to scala tympani by a hole, by a small opening called helicotrema, and the base of the cochlea or base of the scala vestibuli. Base is made up of oval window and round window. Oval window is covered by, I've got a foot plate of stepes, and the round window is covered by the secondary tympanic membrane. See, at the base of the cochlea, the scala vestibuli ends at the oval window, and the scala tympani ends at the round window. The basilar membrane is attached medially to the nimbus, or medially to the bony spiral lamina, and laterally to the fibrous spiral ligament. The Richner's membrane is attached medially to the wall of the limbus and laterally to the upper margin of the stria vascularis. Here we can see in the diagram. The thing is, medially, Richner membrane is attached to the nimbus. Vesicular membrane is attached. Basilar membrane is also attached. This is medial portion. This is lateral portion. Richner's membrane is attached to the at the stria vascularis, and this basic membrane is attached to the spiral ligament. So this forms the medial side, and this is the lateral side. These are the three compartments: scala vestibuli. This is the Richner's membrane. This is organ of Corti that lies on the basilar membrane. This is scala mediae. This is scala tympani. This is nimbus. See, the basilar membrane is medially to the bony spiral lamina and laterally to the fibrous spiral ligament. The Richner's membrane is attached medially to the wall of nimbus. That is, medially, limbus is there, and laterally there is a upper margin of stria vascularis. Organ of Corti. It is a primary receptor of hearing located on the basilar membrane, extending from the apex to the base of the cochlea. On the basilar membrane, there stand two rods called rods of Corti, which projects into the scala media. In between the two rods lies a tunnel called tunnel of Forti, which is filled with perilymph. Medial to the inner rod, single row of inner cells is present, and lateral to the outer rod, 3 to 4 rows of outer cells are present. They are about 3,500 inner hair cells and 20,000 outer hair cells. Here we can see in the diagram, this is basilar membrane, this is organ of Forti. This from the basilar membrane, the two rods are coming up. This is rods of Forti, and they form a tunnel, that is tunnel of Forti. Medial to the rod of Forti is the medial to the rod of Forti is inner hair cell, and lateral or outer side is outer hair cells. There are 3 to 4 rows of outer hair cell, and medial to the rod is inner hair cell. This is basilar membrane, this is rod of Forti, this is tunnel of Forti. Medially, inner hair cells are present, laterally, outer hair cells are present, and both are supported by the pharyngeal cells. This is outer pharyngeal cells, and this is inner pharyngeal cells. Inner pharyngeal cells. Outer pharyngeal cell are data cells also. Now, inner hair cells are supported by inner pharyngeal cells, while the outer hair cells are supported by the data cells or outer pharyngeal cells. From the upper surface of the hair cell, they project tiny cilia called stereocilia, which pass through the thin, dense granular membrane called as reticular lamina and gets embedded into the tectorial membrane. Here we can see in the diagram. See, this is reticular lamina. So these inner cell, hair cell, and outer hair cell have got cilia that passes through the reticular lamina and get attached to the tectorial membrane. This is tectorial membrane, starting from the nimbus, or attached to the middle part of the spiral this nimbus. Right. Then the stereocilia, which passes through the reticular lamina, are bathed in the endolymph, whereas the base of the hair cells are bathed in the perilymph. The tectorial membrane is a thin, stiff, gelatinous, elastic structure made up of glycoprotein, which is attached to the one end to the nimbus and other end to the attached to the supporting cells or Hensel cells, which lies outside the outer hair cells. Here we can see in the diagram. See, this is tectorial membrane. This is reticular lamina. The stereocilia starts from at the tip of the hair cell. These are processes that is called cilia, and they passes through the reticular lamina and ends up in the tectorial membrane. Tectorial membrane is attached to the nimbus on one side, that is medially, and laterally or outer side it is attached to the pharyngeal cells. Outer pharyngeal cells or Hensel cells, also called as, these are the type of cells, supporting cells, that are Hensel cell, Claudius cells. These are the names of the inner outer pharyngeal cells that supports the outer hair cells. Now the vibration of as the sound reaches, the movement of the foot plate of the steppies it causes the movement of the foot plate of the steppies in the oval window and sets up the pressure in the perilymph of the scala vestibuli. As the sound comes, there is a movement of the foot plate of the steppies in the oval window, and it causes pressure in the perilymph, of the scala vestibuli, the basilar membrane is readily get depressed into the scala tympani by the pressure. Here we can see the diagram. See, as the sound reaches, footplate of the stapes moves inside, it put pressures, and this retinous membrane, it presses downwards and also leads to the movement of the basilar membrane downwards. So you can have a idea. Now, the basilar membrane is readily get depressed into the scala tympani by the pressure. Finally, the pressure is transmitted by scala tympani to the round window, causing the secondary tympanic membrane to bulge outside in the middle ear. Now, the top of the hair cells in the organ of aorta are held by the rigid reticular lamina, and their cilia are embedded into the tectorial membrane. Discussed already, the tectorial membrane and basilar membrane are attached to the nimbus. Therefore, when the basilar membrane moves, there is a sharp motion between the tectorial and reticular lamina, causing the bending of the processes of the hair cell. And this bending of the processes will lead to the generation of action potential. See, in this diagram we can see this is nimbus, this is tectorial membrane, these are the stereocilia, these are the hair cells, inner hair cell, outer hair cell, rods of corti, tunnel of corti. Tunnel of corti is filled with perilymph, and this tectorial membrane is in the scala media and contains endolymph. So the processes are bathed in the endolymph, and the bases of the hair cells are bathed in the perilymph. Now, as this stereocilia move, what happens when the basilar membrane moves upward? Basilar membrane moves upward, it will lead to stereocilia to move away from the nimbus. As the basilar membrane moves upward, it will lead to stereocilia to move away from the nimbus, and this causes opening of the potential channels, and the cell gets depolarized. And if the basilar membrane moves downward, the stereocilia moves towards the nimbus, and this will lead to the closing of the potassium channels, and that will lead to the hyperpolarization. Here we can see when the basilar membrane moves upward, the stereocilia moves away from the nimbus, causing the opening of the potassium channel, and this leads to the depolarization of the hair cell. When the basilar membrane moves downward, the stereocilia moves towards the nimbus, causing closing of the potassium channel, and this will lead to the hyperpolarization of the cell. See, this is nimbus, this is tectorial membrane, this is stereocilia. When the basilar membrane moves upward, it leads to the movement of the stereocilia away from the nimbus. It is seen away from the nimbus. This will lead to the opening of the potassium channels and will lead to depolarization. Here, where the basal membrane moves downward, the stereocilia moves towards the nimbus. This is nimbus, it moves towards the nimbus. Now, electrical activity of the cochlea, if there are two electrodes are inserted in the scala media containing endolymph, and other is inserted in the scala vestibuli containing perilymph, and these electrodes are connected to cathode ray oscillograph through suitable amplifier, a steady potential difference of 50 to positive 50 to positive 100 millivolt is recorded. This potential is called as endolymphatic potential or endocochlear potential. As we know that the Richness membrane separates two fluids, perilymph in the vascular vestibuli and endolymph in the scala media. The endolymph is formed by the stria vascularis and has an electrolyte concentration very similar to interstitial cellular fluid, intracellular fluid, while perilymph is formed mainly from the plasma and has got electrolyte concentration very similar to extracellular fluid. This is the difference because there is a difference in the potassium ion. The processes of the hair cell projects into the endolymph, whereas the bases are bathed in the perilymph. This arrangement is necessary for the production of generator potential. Hair cell depolarization initiates the release of chemical transmitter glutamic acid, that is glutamic acid, that activates the receptor site on the terminal of the afferent neuron and causes the generation of action potential in the neuron. Now we come to the diagram. This diagram. See, this is basilar membrane. This is Richness membrane. Richness membrane separates scala vestibuli from scala media. Scala vestibuli contains perilymph. Scala media contains endolymph, and basilar membrane divides scala media from scala tympani. Scala media contains endolymph, and scala tympani contains perilymph. On the basilar membrane is the presence of organ of Corti, and from the basilar membrane, two rod-like structures move upwards, and these are called as rod of Corti, and they got a tunnel in between. This is called tunnel of Corti, and this is filled with perilymph. Then this limbus is medially and stereovascular is laterally. So the medial to the medially, the inner hair cells are present. That is, this is the inner hair cell, and laterally outer side, the outer hair cells are present, and these outer cells are supported by outer phalangeal cells, and inner hair cell is supported by inner phalangeal cells. This is tectorial membrane, and these are the stereocilia arising from the inner hair cell and outer hair cell, and this is tectorial membrane. This is tectorial membrane. This is reticular lamina. Reticular lamina. So the processes of the hair cells passes from the reticular lamina and ends up into the tectorial membrane. These processes are bathed in the endolymph because this is situated in the endolymph, that is scala media. Fluid in the scala media is endolymph, and so the inner hair cells are bathed in endolymph, and bases of these cells are bathed in. See, these are the bases of this are bathed in perilymph. That's why this is responsible for the generation of action potential when the basilar membrane moves. See, when the basilar membrane moves upward, it will lead to the movement of stereocilia away from the nimbus. This is the nimbus, and this will lead to the opening of potassium channels and will lead to the depolarization of cell. And when the basilar membrane moves downwards, it causes the stereocilia to move towards the limbus. It causes closure of the potassium ions, potassium channels, and that will lead to the hyperpolarization of the cell, hair cell. Now this hair cell depolarization initiates the release of glutamic acid that will activate the afferent neuron. That means that it is afferent neuron of the cochlear division of the eighth nerve. Now we move to auditory pathway. Auditory pathway is a five-order neuron system. The first order neuron consists of cochlear division of the eighth cranial nerve that starts from the hair cell. Cochlear division of eighth cranial nerve starts from the hair cells and project to the ipsilateral, dorsal and ventral cochlear nuclei of the medulla. See, in this diagram, we see that this cochlear division of a cranial nerve is coming from the inner hair cell or from the hair cells, and they ends up into the dorsal and ventral ipsilaterally. Dorsal, this is dorsal cochlear nuclei, and ventral cochlear nuclei, this is cochlear nerve. They ends in ventral cochlear nucleus and dorsal cochlear nucleus ipsilaterally. This is first order neuron. Then the second order neuron starts from the cochlear nuclei and projects into the superior olivary nucleus and trapezoid body, bilaterally and contralaterally, respectively. See, here we can see these are the from the dorsal cochlear nuclei and ventral cochlear nuclei. The fibers goes to the upwards. They ends up into the superior olivary nucleus and trapezoid body. The fibers go bilaterally also and contralaterally also to the trapezoid body. See, there's a dense innervation that leads to the appreciation of the or discrimination of the sound, or high discrimination of the sound can be possible. So these are the second order neuron, and then third order neuron fibers. Third order neuron fibers arises from the olivary nucleus, superior olivary nucleus, and ascends up bilaterally in the lateral lemniscus to end in the inferior colliculi of the midbrain. And fibers from the trapezoid body ascend ipsilaterally in the lateral lemniscus to the inferior colliculi. So the fibers, third order neuron starts from the superior olivary nucleus and trapezoid body, and they ends up into the inferior colliculi of the midbrain. The fourth order neuron projects bilaterally from the inferior collicular nuclei to ends in the medial geniculate body of the thalamus. And fifth order neuron starts from medial geniculate body and terminates in the primary auditory cortex, area 41 and 42, situated in the superior temporal gyrus. See, this is cochlear division of eighth nerve coming from the inner hair cell and outer hair cells. They ends up into the dorsal and ventral cochlear nucleus in the medulla. These are the first order neuron. From here, the fiber starts and they move contralaterally and bilaterally and ends up into the superior olivary nucleus and trapezoid body. This is nucleus of trapezoid body. This is superior collicular. These are second order neuron, and this second order neuron then moves upwards, passes from the lateral lemniscus and ends up into the inferior colliculi. Inferior colliculus, these are the third order neurons. See, they are going bilaterally and also contralaterally. Bilateral for achieving more discrimination in the pitch sound, intensity of the sound, or more discrimination of the sound. So these are first order neuron, second order neuron, third order neuron, ending up into the inferior colliculus, and from the inferior colliculus, fourth order neuron starts. They ends up into the medial geniculate body, and from the medial geniculate body, fifth order neuron starts. They end up into the primary auditory cortex, area 41 and 42. So again, to repeat, that the cochlear division of eighth nerves, they ends up into the dorsal and ventral cochlear nucleus. This is first order neuron. Then fibers move upwards and ends up in the superior olivary nucleus and trapezoid body. These are the second order neuron, and the third order neuron starts from superior olivary and trapezoid nucleus body and passes through the nucleus of lateral lemniscus and ends up in the inferior colliculus. These are the third order neuron, and from the inferior colliculus, the fibers they extend in, they move upwards and ends up into the medial geniculate body. These are the fourth order neuron, and from the medial geniculate body, the fibers ends up into the primary auditory area or primary situated in the temporal gyrus. Situated in the superior temporal gyrus, this is the area 41 and 42. So this is auditory pathway.